right. Good afternoon, everyone. Thank you for having us here. And my name is Kalen Hall.
And I'm Leo Williams, and we're the co-founders of Informuta. So our mission is to reduce the costs and lives lost to antibiotic resistance. And so we're developing novel diagnostics for antibiotic resistance using machine learning.
So the WHO, or the World Health Organization, predicts that antibiotic resistance will overtake heart disease as the global #1 cause of death by 2050, taking 10 million lives a year. But already this problem plagues the hospital system, with over 1.4 million resistant hospital-acquired infections occurring annually that result in $70 billion in direct losses every year. Choosing a first effective antibiotic treatment is critical to curbing this crisis. But the problem is that the current standard of care forces clinicians to do what's called empiric treatment, which is essentially like an educated trial and error, rather than basing their treatment plans on hard data. So, next generation sequencing, or NGS, poses a promising new method and new solution as it's more sensitive, more specific, and more accurate than the standard of care. But the problem is that there's no way to diagnose antibiotic resistance currently available based on this NGS data.
So to solve this, we're developing a precision diagnostic service to analyze antibiotic resistance. So first, we use next generation sequencing data to analyze the whole genomes of bacteria from clinical infections. Then with our novel machine learning pipeline, we analyze the DNA for patterns of mutations, which allows us to vastly increase the accuracy, thus eliminating our reliance on individual mutations in the bacteria. So this allows us to diagnose infections that are currently resistant, but it also allows us to be the first to diagnose infections that are resistant prone, will develop resistance. In addition, it allows us to track patient infections in real time for hospitals. So we take all this and compile it into a digital report to deliver to physicians within 2 hours of receipt of the raw data. Our MVP will be ready by December, and just want to know we plan for this to be the first, let's say, broad spectrum molecular antibiotic susceptibility test that is species independent. So in addition, secondarily, we plan to build and maintain the largest bacterial genomic database, which we will use to retrain the models informing our diagnostic, continuously analyze for new biomarkers, and use as basically a foundation for future product development. So in talking to clinicians so far, they say they're most excited about being able to do just one comprehensive test to eliminate guesswork. This allows them to use targeted treatment, which makes the first round of treatment much more effective and reduces the tendency for antibiotic misuse. They also like that it's trained on direct patient samples and not data that's grown in test tubes in the lab, which can alter the phenotype of clinical infections. And it also allows us to help them track real-time infection spread to control outbreaks early. In addition, they like that it reports our novel resistance prone metric, which really helps them to decide better courses of treatment. So all of these are very vital for managing chronic infections and long-term hospital stays, especially in populations such as those with diabetes, organ transplant patients, and otherwise immunocompromised patients. So we believe that our diagnostic will increase the treatment success rate, thereby decreasing the overall cost of treatment. In turn, decrease the overall cost of treatment by decreasing the patient length of stay and also decreasing these these penalties that hospital incur due to mismanaged infection treatment. So our services also help prevent emergence in the first place by really focusing on the effectiveness of that first round of treatment. And all of this really boils down to providing value to hospitals through improved patient metrics and reduced monetary costs.
So comparing our services to basically what hospitals use routinely now. So most hospitals use either of two kind of base tests. It's either phenotypic based test, meaning that they grow, then isolate out from the infection sample, and then they test it against antibiotics in a plate or a test tube. The other one is PCR based. So they basically do a screen for a couple of known mutations to see if they get a hit for any resistance markers. So we are the only service that is based on next generation sequencing data. So that has pretty big advantages compared to phenotypic based testing. So kind of like Leo was mentioning, that phenotypic based testing does not always project how well a drug will do when you actually treat the patient with that. It's also significantly slower. It can take anywhere from 4 to 7 days to get results back, which by then they're not really clinically actionable. Compared to PCR-based testing, we do not rely on specific mutations in the NGS data, whereas those PCR-based tests kind of have anywhere up to 5 to 20 known resistance markers that they're screening for. But if there's a resistance marker that's not in that panel and that infection has it, you're going to miss it and misdiagnose. So in addition to all these advantages, we're also the only services to report the resistance prone metric, and we're also the only service to do simultaneous infection spread tracking for hospitals. So we plan to market this as a software as a service to both private clinical diagnostic labs that are based on next generation sequencing and also to large academic medical centers that have NGS capabilities. We basically just charge a fee per service for every test, and then our pricing follows a tier-based system based on how complex the analytics will be. So the market for in vitro infectious disease diagnostics, which is what we'd be considered right now, is growing at a CAGR of 6% to reach $107 billion by 2028. The market for NGS-based testing is growing even faster at 22%. So our total addressable market, just with these diagnostic services that we are offering, we are planning to offer now, is $1.2 billion. And if we can service just 10 hospitals, we'd be making an annual revenue of $18.7 million. So we've already extensively established our proof of concept data, both on lab strains and clinical isolates. We filed a provisional patent back in February, which will convert to 2 PCTs February 2024. And we're currently now doing R&D to build up our full pipeline, and then focusing on clinically validating it on patient samples, which will be complete by early next spring. We plan to file for FDA exemption first, which will allow us to hit the market selling to private labs as a diagnostic tool. And then we'll get the data from this testing to then accumulate, build up, and file for an FDA 510(k) clearance that will license us as, or allow us to be a software as a medical device, which will make it easier for us to directly sell to hospital systems.
So right now we're currently seeking to raise $1 million in pre-seed stage funding to last us through all of next year, with the primary end goals of being able to conduct a prospective multi-center observational clinical trial and also obtain our first customers and our first revenue. So we think this amount will get us all the way to market.
So our team consists of myself. I'm Kalen Hall. I'm a 4th-year PhD candidate in Tulane's Biomedical Sciences program. My dissertation research focuses on understanding the genetic pathways of how bacteria acquire multi-drug resistance, with a very heavy emphasis on next-generation sequencing, analyzing mutations, and assessing multi-drug resistance.
I'm Leo Williams. I just recently defended my dissertation in Tulane's Bioinnovation program, where I studied DNA damage and repair in cancer, and also how to apply computational methods to study mutagenesis.
And we're advised by a team of mentors with expertise ranging from bacteriology, genetics, computational biology, and entrepreneurship. So we're Informuta again, and we hope that with the development of our diagnostic services, all patients with bacterial infections can receive personalized, precise, and effective treatment so that we can mitigate the global health crisis of antibiotic resistance. Thank you guys so much for your time, and we'd be happy to take any questions.
So just 2 quick questions, or 2 parts to the question. First, how many mutations do you typically see, just as a general idea, in each bacteria that become resistant, and how accurate are the typical PCR panels for predicting that?
Sure.
So it can be anywhere from dozens to hundreds. It's really dependent on what kind of reference data you're using. That's something we plan to investigate also. And then how does it compare?
Yeah, like kind of going off what Leo was saying. So when we do this analysis, it really depends on what reference genome you're calling mutations to. So that can really drive how many mutations you get. So that's a big problem that we're also solving, is getting a good reference genome. In terms of the PCR tests, I don't know the accuracy off the top of my head. I know they work pretty well. Yeah, from all the clinicians we talked to, they say they work well. It's kind of a multifaceted problem of the speed, time, how well the tests predict how the drugs are going to work. And especially when we talk about for those chronic infections and long-term hospital stays, I think those are kind of where those tests tend to fail, and they need to order anywhere from 5 to 10 tests to actually get an actionable result.